In today's video at Medical Sciences, we want to discuss about the structures of proteins, whereby under classification of proteins we see classification by function, classification by structures. So we want to look at classification of proteins by structures. And as we begin, we urge you to subscribe if it is your first time. You can subscribe, then like the video, then share to many so that we can continue to grow. So among the structures of proteins, we are going to discuss about the primary structure. Then we shall look at the secondary structure, tertiary, and then quaternary structure. And in each, we are going to discuss how are the bonds formed and different types of bonds and examples. So we're going to see primary structure. And this primary structure of protein is, here we are going to look at a linear polypeptide chain. So it is composed of a linear polypeptide chain. This is composed of a linear polypeptide chain. And this linear polypeptide chain is formed due to the type of bond. The type of bond in this linear structure is called the peptide bond. So we are seeing the peptide bond holding this linear structure of different amino acids. So we have many amino acids being joined together by a peptide bond between the carboxyl group and the amine group of the incoming amino acid. So like if we have a structure, like if we have different amino acids, so a linear polypeptide chain we are talking about is finding this one made up of many amino acids. So these are the amino acids, and these amino acids which are on this chain, these are the amino acids, and the bond that is joining them is what we are calling the peptide bond. And this peptide bond, if I can illustrate some structure of the amino acid, whereby here we have the allyl group, the hydrogen, then we have the carboxyl. So we are going to see this carboxyl group joins with the hydroxyl group is lost. Remember, it has the hydroxyl group, it is lost, and it joins to another amine group, then another carboxyl group. So what we call a peptide chain is this, the bond formed between the carboxyl group and the amine group of the incoming amino acid. So this is what we regard to as a peptide bond. Another one, we can put it here that at another carboxyl group you can join another amino group. This is the variable chain, this is the hydrogen, then we get another carboxyl group. So this one becomes another peptide bond. So these are the peptide bond formed during this linear structure which we are discussing with. So one thing we have to note under primary structure, it is the basis of all other types of proteins, whereby this is a linear polypeptide chain, whereby it has numerous amino acids composed of many amino acids, and these amino acids are joined to one another via peptide bond. So the only bond found in the primary structure is known as peptide bond, formed between the carboxyl group of the first amino acid with the second incoming amino acid, and they continue to form like the other. The way we have seen these two being formed to form a linear polypeptide chain of amino acids. So this is what we can discuss about the primary structure of proteins. Then the second structure we can talk about in this session is the secondary. We can discuss with you the secondary structure. We have secondary structure, and under secondary structure, here we are going to see the polypeptide chain formed in the primary structure is going to undergo twisting and folding. So we are saying that secondary structure is formed by twisting and folding of a linear polypeptide chain. So if we have a linear polypeptide chain formed during the primary structure, it undergoes folding and twisting to form a secondary structure. So it is formed by twisting and folding of a linear polypeptide chain, which is the primary structure. And this chain, after folding and twisting, it is the one that results into formation. So the folding and the twisting results into formation of the alpha helix. Results into formation of alpha helix, and also results into formation of beta pleated sheets. We form beta pleated sheets, and these beta pleated sheets can either be parallel, they can be parallel, or they can be anti-parallel. So this is what we can see under secondary structure. Further folding and twisting of a primary structure, that is a polypeptide chain, via hydrogen bonding. Here the type of bonding that is formed during the secondary structure is the hydrogen bond. So the folding and twisting of the polypeptide chain is due to the interactions of the hydrogen bond in between them. And this hydrogen bond can either be intramolecular or intermolecular, the way we are going to see under alpha helix structure and beta pleated. Alpha helix has intramolecular hydrogen bond, whereas beta pleated sheets have intermolecular hydrogen bonds. So an example of a beta pleated sheet of we are going to begin with alpha helix. So this is the structure of the alpha helix, and this alpha helix structure, it is going to have, so this is the secondary structure. This is the formed due to folding and twisting due to interactions of the hydrogen bond. That if we have a carbonyl group at this point, we have, this is the first one, we have a second carbonyl compound, we have a third carbonyl compound. So this one, which is the first, then this is the second, this is the third, this is the fourth, they are going to interact with the fifth. This one is going to interact with the fifth amine group, the hydrogen on the amine group, to form what we call the hydrogen bond. So this one, which is in the second position, interacts with also another amine group to form hydrogen bond, and this one can interact with another amine group here to form what we call hydrogen bond. So this hydrogen bond formed in between, we call it intramolecular. This hydrogen bond which is formed, we regard it to as intramolecular hydrogen bond. Intramolecular hydrogen. Why do we call it intra? Because this hydrogen bond formed in this chain is within the chain. It is not joining the two. We have only one chain from N terminal to C terminal. N terminal, that is where the amine group is, and the C terminal is where the carboxyl group is. So if it is 1 and we are forming in it, due to folding, we are forming hydrogen bonds, is what we could regard to as intramolecular hydrogen bond. So this is also another intramolecular hydrogen bond. So in summary, what forms alpha helix in secondary structure is the intramolecular hydrogen bond. So the type of bond is intramolecular. Then another structure we can see under secondary, we have alpha helix as one of the structure. Then the second structure we can talk about is the beta pleated sheet. And we said under beta pleated sheet, we can have parallel. Parallel is when all of them are in the same direction. We are seeing this one from N terminal to C terminal. Even this is from N terminal to C terminal. So these are parallel. They are all moving in the same direction. So this is what we call parallel beta pleated sheets. So we can name it parallel beta pleated sheet. So this parallel beta pleated sheet, which is formed during the folding and twisting, we are seeing this is the first polypeptide chain. This is the second polypeptide chain. And we are seeing the two polypeptide chains. These are two primary structures. One primary structure and another second primary structure. So when we join them via hydrogen bonding, for example, if we have the carbonyl here, then here we are going to have the R group, then we are going to have another amine group followed by the carbonyl group. Then we are going to have another one here. This one is N terminal. So this one becomes, so we're going to see the N is going to be here, hydrogen bonding. Then here we're going to have, so we're going to have the amine here. Then we have the carboxyl here. We're going to have the amine here. Here we shall have R group. Then we shall have another amine group and so forth. So during bonding, the bonding occurs between the carboxyl group and the amine group. So this carboxyl group is going to bond with the amine group here. And this amine group here is also going to bond with this carboxyl group. So we can appreciate that these intermolecular hydrogen bonds, they are not stable because they are not straight, because all of them are moving in the same directions, so they are bending, and then they don't form a stable structure of beta pleated sheet. So these are the ones we are calling intermolecular hydrogen bond. Call them intermolecular. Hydrogen. So in beta pleated sheet, we don't have intra. Here we had intra, here we have inter, because we are joining two polypeptide chains to form unstable. So the one formed by the parallel beta pleated sheet is unstable. It needs to be stabilized. And it is formed whereby all the polypeptide chains, two of them, are moving in the same direction. And then we see the bonding of the hydrogen between the carboxyl group and the amine group of another polypeptide chain, forming intermolecular hydrogen bonding. So this is what we can talk about beta pleated sheets parallel. Then the last one we have antiparallel. This one is anti-parallel beta pleated sheet. And this anti-parallel beta pleated sheet, for it, the two polypeptide chains are anti-parallel. They are moving in different, or what we call opposite direction. This one is moving from C terminal. This is moving from N terminal to C terminal. Then this one is also moving from this direction towards this direction. N terminal, then to C terminal. So they are moving in opposite direction, whereby this one is from N terminal to C terminal, then this one begins also in the opposite direction. So they are moving in the opposite direction. That's why we call them anti-parallel beta pleated sheets. And these beta pleated sheets, they form straight hydrogen bonds. That if I have a carbonyl group here, it's going to interact with the hydrogen at the amine group to form a straight hydrogen bond. We are going to see another carbonyl group here combining with the hydrogen group to form a straight hydrogen bond. And we're going to see another straight hydrogen bond. If we have a hydrogen here and a carboxyl here, we're going to form a straight. So this one forms straight hydrogen, intermolecular hydrogen bond. Straight intermolecular. Inter because it is joining two different polypeptide chains, so that's why we call it intermolecular hydrogen bond between these two polypeptide chains moving in a different direction to form a stable intermolecular hydrogen bond between carboxyl and the amine group. So this one is very stable compared to the parallel beta pleated sheet in the secondary structure. So in summary, under secondary structure, you have to tell us that the primary structure which we formed, it undergoes more folding and twisting to form a polypeptide chain, to form the chain which do via hydrogen bonding, whereby this folding and twisting by hydrogen bond results either into alpha helix with intramolecular hydrogen bond or beta pleated sheets with either parallel or antiparallel joined together by intermolecular hydrogen bonds. And this is what we have illustrated, whereby this is the structure of alpha helix, then the structure of the parallel beta pleated sheet, then the structure of the antiparallel beta pleated sheet of proteins. So after finishing secondary, we are now going to look at the tertiary. Because these secondary structures we have formed, the beta pleated sheets and the alpha helix, they can undergo further folding and twisting. So we are seeing the secondary structure formed whereby we had the alpha helix and the beta pleated sheet. These two, they undergo further folding. They undergo further folding and twisting to form a tertiary structure. To form a tertiary structure. And how is the tertiary structure formed? What causes the further folding and twisting? This one is formed by bonds which include hydrogen bond. It includes ionic bond. It includes covalent bond. It has disulfide bond. And it also involves van der Waals forces. So these different bonds, that is hydrogen bonding, ionic bonding, covalent bonding, disulfide and van der Waals forces, they are the one that brings about further twisting and folding of these secondary structures to form a tertiary structure. And this tertiary structure which is formed is a 3D dimensional structure. Is a 3-dimensional structure. So in illustration, we are going to have a secondary polypeptide chain undergoing further folding and twisting to form a tertiary structure. So this one which is formed is known as a tertiary structure with a 3D dimension, meaning it has height, length, and width. Three-dimensional structure. So it is due to further folding and twisting due to interaction of the hydrogen bonds, ionic bonds, covalent disulfide bridges, and van der Waals forces, which forms this tertiary structure. And this tertiary structure formed is catalytically active. This structure is catalytically active. It is catalytically active. And example of a tertiary structure, we can give myoglobin. So example here, we are giving myoglobin and some enzymes which are catalytically active. So those are the example. Myoglobin belongs to the tertiary structure. So as we are looking at the conclusion, we are now going to look at the last structure, which is known as quaternary structure. And this quaternary structure, you see, we are moving whereby we have seen the primary structure giving rise to secondary structure giving rise to tertiary. So the tertiary structure are also going to give us what we call the quaternary structure. And under quaternary structure, we are going to see this one occurs in proteins. It occurs in proteins which are polymeric. These proteins which have quaternary structure, they must be polymeric. They have to be having different polymers or different polypeptide chain. Because we are seeing the tertiary structure is formed by a monomeric. Monomeric meaning it has one polypeptide chain. But these quaternary structures, they are poly, meaning they have more than one polymer. And examples of these proteins which exhibit quaternary structure, we have hemoglobin. Hemoglobin is one of them. Another enzyme like lactate dehydrogenase, whereby it is for it, this hemoglobin has 4 polymers. This one has 4 polymers. Then LDH has 5 polymers, so it is the pentameric. So this one is the pentameric because it has 5 polymers. This one has 4 polymers. And these polymers, which are different, like hemoglobin with 4 polymers, they are being joined together by different bondings. And these bondings we are going to see in this video, they are the same for tertiary. The bonds which join these polymers together, still we have the hydrogen bonds. We have those which are covalent hydrogen and peptide bond. Then the non-covalent, these are covalent. Then non-covalent bonds, we are going to see examples of ionic bonds or ionic interaction. We are going to see disulfide bridges. We are going to see van der Waals forces. All these bonds, the covalent and the non-covalent, bring about formation of either trimeric, quaternaric, or pentameric, or even hexameric. The way we have seen hemoglobin with 4 polymers and LDH with 5 polymers. Whereas another example of enzyme is creatine kinase, which has 2 dimers. It is dimeric because it has 2 polymers. So an example, I can draw an example of hemoglobin, whereby hemoglobin can have 1 polymer, then it can have the second polymer made up of a single polypeptide chain, then it can have another third and another fourth polymer. So it has 4 tertiary structures which are monomeric, and these different forms which are here, one polymer, then another different polymer formed, then another different polymer here, and another different polymer. They are joined together by these bondings, whereby this bonds with this bonds with this bonds with this, to form a single structure which is under quaternary structure. And that is how hemoglobin appears to be under quaternary structure. So we have, in this video, we have discussed about different things. We have seen different structures from primary to tertiary, and in each structure we have known what is it, the type of bond, and the examples. Under primary, we have seen there it is a linear polypeptide joined by the peptide bond to form a linear polypeptide chain between the carboxyl group of the first amino acid with the second incoming amine group. Then we have seen secondary. If the primary folds further by hydrogen bonds, it forms what we call alpha helix and beta pleated sheets. And this hydrogen bond we have seen, it can either be intramolecular, that is in alpha helix, or intermolecular, that is in a parallel beta pleated sheets and parallel beta pleated sheets, which forms stable hydrogen intermolecular bond. Then we have seen that if these structures of secondary, the alpha helix and the beta pleated sheet, undergoes further folding and twisting due to the hydrogen bond ionic interactions, the sulfide bridges, we form a catalytically active protein, which is a 3-dimensional structure. And examples we have given is myoglobin and some enzymes. Then we have ended up with the quaternary structure, which is found in the proteins that are polymeric, having different polymers. Some can have 2, like creatine kinase, others can have 4, like hemoglobin, then another one can have 5, like LDH, and so forth. And we have seen they are joined together by different bondings, the sulfide bridges, ionic van der Waals forces, hydrogen bond, and peptide bond. Thank you so much for learning with us. Hope you have gained a lot from this video. May God bless you.